Can you see my screen now?
Yes.
Okay. So this is week 6 TA session for biointerface engineering. Let's see what all topics were covered in this week. So lecture 16, lecture 17 and lecture 18 were the 3 lectures that were covered in week 6. Lecture 16 had topics on host response to biomaterials, cell surface interaction, complement pathways, sequence of responses. So basically, we mostly need this when we are planning to introduce biomaterial to our body. So when the body is exposed to this foreign material or biomaterial, anything that is uh, not part of our body is foreign. So these biomaterials are also foreign materials. So when they are introduced to our bodies, they have a different response. So cell surface interaction, cell surface interaction is what we need to look into when discussing about this host response to biomaterials. So when a implant or a biomaterial is exposed to our body, it comes in contact with water first and then different proteins, and then these proteins then attract cells. And mostly the outer part of cells interact with this biomaterials. And the interactions are in 3 phases. First, adhesion. The cells adhere on the surface of the biomaterial. Adhesion. Next is proliferation, wherein the cells start to divide and proliferate. Adhesion, proliferation. The next one is differentiation, wherein the cells will differentiate to perform a specific function. Differentiation. And these events from adhesion to proliferation to differentiation occurs over a period of days to weeks. Next in the lecture, we also we come to discuss that host cell's response is mainly because of two reasons. One, implant or the biomaterial, and second is tissue injury, which mainly happens when trying to implant this in the body. Tissue injury. So the tissue injury, it mainly follows a normal sequence of events that our body follows when it undergoes a injury, that is the wound healing process, which gets resolved soon after. But then the implant will lead to, this is mainly wound healing. This is wound healing, and this will lead to chronic inflammation. Of course, it depends on the material's reactivity to the body as well, but most of the time, because it is not moving anywhere, it's still in contact with the body, it will lead to continuous, persistent inflammatory response. Next, discuss this wound healing. There is a sequence of processes in this wound healing, and these inflammation or the damage, the degree of to which how much this tissue injury or the implant uh, disturbs the homeostatic mechanism of the body actually corresponds to the host's response to the biomaterial. So the body homeostasis follows this sequence. First is vascular spasm. So during tissue injury, there is damage to the blood vessels, and then the blood vessels constrict and they increase the flow of blood to the site of injury. And then there is formation of platelet plugs. Formation of platelet plug. So as soon as the blood rushes to the site of injury, it also carries with it platelets, leukocytes, and all kind of immune cells as well. And next, this these cells then activate coagulation cascade. They activate a coagulation cascade, wherein different enzymes, clotting factors, those take part. So in this step, thrombin acts on fibrinogen. Thrombin acts on fibrinogen to convert them into fibrin molecules. These fibrin are insoluble and they participate in the formation of fibrin clots or a fibrin plug. And the end step would be then fibrinolysis, where this fibrin clot is dissolved. So these were the sequence of wound healing process that occurs during any tissue injury. And then also there are complement pathways. So when a foreign material, or when anything is when our body experiences a response event other than a regular event, these complement pathways are activated. There are three different complement pathways. One is classical, next is lectin, third is alternate pathway. Alternate pathway. So the classical pathway is initiated due to the antigen antibody complex. The lectin pathway is activated due to presence of carbohydrate binding domains due to pathogen or any other uh, event. And then the alternate pathway occurs due to exposure to pathogenic surface or foreign materials. Or foreign materials. So what we are really interested in this is the alternate pathway, because that is most relevant to what we are trying to look now. So when our body is exposed to this implants or foreign materials, this alternate pathway is activated. and it initiates a cascade of event, and actually all these three pathways converges at C3 and C5 convertase. C3 C5 convertases C3 C5 convertases, and then they lead to different response such as inflammation, lysis, and opsonization. So next, so basically these cascades are activated due to different kind of events, and then they convert at C3 C5 convertases and lead to inflammation, lysis, and opsonization. Next in the lecture we discuss about the sequence of responses. So during this exposure to foreign materials or whatever we have been discussing so far, there is a sequence of events that take place during this implantation of foreign material in the body. So the sequence is as follows. First, injury, as we were discussing. You need to create an injury to insert an implant. And this injury then will lead to acute inflammation. There is acute inflammation followed by chronic inflammation, and that follows granulation tissue. Most of the cells will start adhering and denaturing over the surfaces, and it will cover the surface and it will form a granulation tissue. This will elicit a reaction from the host body, and at the end fibrosis occurs. Fibrosis occurs. So basically, if things go wrong, if this is the implant, when it is introduced in the body, these sequence of event will take place, and then it will be covered by proteins, denatured proteins, which will attract cells all over it. Proteins and cells. These cells then secrete more inflammatory signals and secrete more proteins, carbohydrates, glycoproteins, and all of their secretions, the denatured proteins and the cells cover the entire implant to stop its further exposure to the body, because it is a continuous state of exposure to the body that is creating problem and eliciting inflammatory reaction. So since the body cannot remove the implant, it will try to cover it with all these secretions and proteins, and it will form a fibrous capsule, and this event is also called as fibrosis. Also, briefly, the factors that affect the cell surface interaction were discussed. For example, the implant, the composition, the mechanical properties, surface topography, their degradability, ability to uh, ability to start infection, resist infection, pro and pre-surgical placement of these. All of these factors contribute to the cell surface interaction of these implants in the body. So this was all covered in lecture 16. We then come to lecture 17. So lecture 17 discusses about the cell adhesion, and as we discussed, when the implant is exposed to the body, the cells adhere on the surface. So there is a mechanism by which the cells adhere on the surface. There are different cell adhesion molecules, work of adhesion, the cell adhesion kinetics. All these things were discussed in lecture 17. So the cell adhesion is basically elicited by the cell surface receptors. So there are receptors on the surface, or basically the cell membrane. These receptors are what These receptors are primarily responsible for this adhesion and response or interaction with the implant. So these receptors are intermediate or intermediate proteins, basically intermediate or embedded proteins, which interact with the implants, and they can lead to different adhesion sites, such as focal adhesion, when the distance between these two is very less. Close contacts, distance is slightly higher than the focal adhesion. And then ECM contact. ECM or extracellular matrix is basically the secretions of the cells, or which basically contains proteins, glycoproteins. And these interact with the implant. So these are the three adhesion sites, and we also discuss about different adhesive proteins, laminin, fibronectin, etc. And then we come to cell adhesion molecules. So there are 5 different classes of cell adhesion molecules. If you want to talk about, that is cadherins, immunoglobulin G, mucin, integrins, and selectance. And then we discuss about integrins, which mostly takes part in the cell adhesion activities. So these integrin is a protein that has different receptor domains for binding to different things, such as RGD for cell adhesion. RGD is basically a 3 amino acid sequence. RGD, arginine, glycine, and yeah, so it interacts with sequences having arginine, glycine, and aspartic acid. If this amino acid sequence is present, then integrin can bind with it because of its receptor domain. Similarly, there are other receptor domains to bind to different sequences and different sequences. And next in the lecture was discussed about the work of adhesion. So different intermolecular interactions such as the van der Waals and electrostatics, those forces are involved and they lead to work of adhesion. So the work of adhesion or the spreading of B over A is given by surface tension of A minus surface tension B minus surface tension AB. And then the Gibbs free energy of adhesion or the delta G of adhesion for this delta G of adhesion or the spreading of B over A or the work of adhesion. If it is less than 0 or if it is negative, then it leads to spontaneous adhesion. Spontaneous adhesion. And then also the characterization of cell adhesion, which can be done by morphological analysis or biochemical analysis, was discussed. The last topic that was discussed was the cell adhesion kinetics. So the cells in bulk NB with the rate of film transfer coefficient interact with N1 plus S which can then go by rate of constant for adhesion Ka, or rate of cell detachment, or it will form cells, where S is the surface here. So all these things were discussed in lecture 17. And then in lecture 18, biocompatibility of the materials were discussed. So the first thing we want when we are exposing our body to these foreign materials is we want them to be biocompatible. That is the ability of the material to perform with an appropriate response in a specific application. So the response that we saw before, whether that is appropriate, so we want that the response is appropriate in terms of what application we want. And that is what decides the biocompatibility of the material. Basically, we don't want an acute or chronic inflammation. Acute or chronic inflammation. We don't want the materials to prevent differentiation of cells. Differentiation of cells. So the when exposing to foreign materials, these are the things, and some more things like this we need to take care of. Now, to assess the biocompatibility of a materials, we can do it either in vitro, that is using a cell line, or this experiments are done out of the body, or we can do it in vivo. In vivo experiments are done with the model, mostly model animals, and they are exposed to the biomaterials and their response is seen. So in vitro we can do so by checking the cytotoxicity, cyt, uh, cytotoxicity, the leachability, or hemocompatibility. Hemocompatibility. And in vivo, we can do so by analyzing the tissue at the site of the implant by performing histology on it. Histology or histochemistry, immunohistochemistry, expression of specific antibodies in that tissue, morphological analysis using transmission electron microscope or scanning electron microscope, and some other techniques such as biochemistry or mechanical testing of the materials. So these are the techniques that we can do to assess the biocompatibility of the materials. Now, now that we covered what all things were taught in this week, lectures in lecture 16, 17 and 18, we'll see the assignment problems for this week. Okay, so now we'll start the assignment problems. Here is the first question. Which of the following sequence of events occur during the cell surface interactions? A. Surface cells, B. Surface water, C. Surface protein. And the options are A, then C, then B, C, then A, then B, B, then C, then A, or it occurs randomly. Which will be the correct sequence of events during cell surface interactions? Okay, so the answer would be option C, B, C, A. First, water will react, then protein, and then cell. As shown in the lecture as well, if this is the implant, first surface water interactions will happen, then protein surface interactions, and then cell surface interactions. Here is the next question. Fibrins are degraded by A. Fibrinogen, B. Thrombin, C. Platelets, or immunoglobulins. What would be the correct answer? Okay, so the correct answer would be thrombin. So during the events that occur during homeostasis, this actually comes into play. So after vascular spasm due to injury, there is formation of platelet plug, and then a cascade, coagulation cascade is activated. Here the thrombin converts these fibrinogen into fibrin. And if you see the actual mechanism, fibrinogen, the thrombin removes this mask over the fibrinogen, and when they are removed, these fibrin monomers then interact with each other to form fibrin protofibril, and then they become long chains insoluble and form the fibrin block. So the answer would be thrombin. And here is the next question. Which of the following is or are involved in cell adhesion? Options are focal adhesion, extracellular matrix contact, close adhesion, all of these. Okay, so the correct answer would be all of these. So there are three different adhesive sites: focal adhesion, cellular matrix contact, extracellular matrix contact, and close adhesion. So when the cell and the surface are in close contact, that is within the range of 10 to 20 nanometer, focal adhesion happens. If the range is between 30 to 50 nanometer, the interaction happens by close contacts. And if it is the distance between the cell and the implant is more than 100 nanometer, the interaction between the cell surface happens by extracellular matrix or ECM contacts. So focal adhesion, close contacts, and ECM contacts, all the options would be the correct. That's why fourth option is the correct answer, all of these. Okay, so here is the next question. Which of the following does not belong to the class of cell adhesion molecules? The options are cadherins, albumins, immunoglobulin G, or mucin-like molecules. Which of these does not belong to class of cell adhesion molecules? Okay, so the correct answer here would be albumin. So there are 5 classes of cell adhesion molecules. Those are cadherins, and then immunoglobulin or IgG, mucin-like molecules, integrins, and selectins. Only albumin is not does not belong to the class of cell adhesion molecules. Okay, now here is the next question. The most common amino acid sequence RGD bind to which cell adhesion molecule? The options are cadherins, integrins, selectins, or Ig superfamily or immunoglobulin superfamily. So here the correct answer would be integrins. So the RGT sequence is basically, these are three amino acids: arginine, glycine, aspartic acid, RGT, and the integrin. This is the integrin molecule, and it has different receptors. So this is basically an RGT receptor. Similar to RGT, it has collagen receptors, leukocyte specific receptors, and laminin receptors. So integrin plays a major role in cell adhesion, and because of these receptors, integrins can bind to RGT sequence. Okay, here is the next question. The percentage of cells adhesion over the surface can be estimated by relating the options are: available surface area for cell adhesion, initial cell number available for adhesion, rate of cell adhesion, or all of these. The percentage of cells adhesion over the surface can be estimated by relating the available surface area for cell adhesion, initial cell number available for adhesion, rate of cell adhesion, or all of these. So here the correct answer would be all of this. So if percentage of cell adhesion over a surface, if the available surface area for cell adhesion, if we know the available surface area for cell adhesion, the initial number of cells available for adhesion, or the rate at which they are adhering or attaching, all these three information can give us an estimate of the percentage of cells adhesion over any surface. Next question. Which of the following criteria is not required for the biocompatibility of biomaterial? A. Cytotoxicity, B. Leachability, C. Hemocompatibility, or D. Ductility. Which of these following criteria is not required for biocompatibility? So here the correct answer would be D. Ductility. So the all the above other options such as cytotoxicity, leachability, and hemocompatibility, these are all in vitro experiments to assess the biocompatibility of the material. And in cytotoxicity basically means to cause toxic effects at the cellular level. So the material as well as the cells are incubated together, and based on depth or alternations in cellular membrane permeability, enzymatic inhibition, based on estimation of these parameters, we can check the cytotoxicity of cells. Leachability is analysis of extracts. So we can extract uh, some of the biomaterials and incubate it with the cells uh, to see uh, if it is releasing any toxic or any, if it is releasing any toxic or danger, any toxic compounds into the cells. Then we have hemocompatibility. This is basically analyzing the cell's compatibility with the blood, and all these tests basically give us an assessment of whether the material of interest is biocompatible or not. Whereas ductility, it is just a property of materials wherein it's just the quality of the material being flexible, that can be bent into a thin wire, which is not at all related with biocompatibility of the material. So ductility will be the correct answer, and it is not required for biocompatibility of biomaterial. Here is the next question. The in vivo analysis of the biomaterial implant involves histochemical analysis, immunohistochemical analysis, biochemical analysis, or all of the above. The in vivo analysis of biomaterial implants involves which of the following? So here the correct answer would be all of the above. So in vivo analysis, we can do histology and histochemistry. This is basically analyzing the tissue, cell tissue of the tissue that is exposed to the biomaterial implants. So the relative number of various cell types and the amount of ECM components around the implant. We can understand this by studying histology and histochemistry. Lysosomal enzymes, alkaline phosphatases, all these things can also give us information about the interaction of the tissue with the implant. Immunohistochemistry, morphological analysis with transmission electron microscopy and scaling electron microscopy, or biochemistry, or mechanical testing as well. So all of these materials, all of these tests can help us in vivo analysis of the biomaterial implant. There is the next question. Which of the following product is generated during the MTT assay? The options are formazan, dimethyl sulfoxide, heteropolymolybdenum blue, or sodium molybdate. So the correct answer here would be formazan. So MTT assay is basically a cytotoxicity assay for in vitro analysis, where we first incubate the cells with the biomaterial and then cell analyze this cell with the help of this MTT assay. So this gives an estimate of metabolically active cells, because the biomaterial may cause some cell death or some cell death. So basically it will give us an estimate of metabolically active cells, and the principle is metabolically active cells produce NADPH dependent mitochondrial dehydrogenase for controlling the electron flux through the electron transport chain. So the assay is based on reducing MTT, which is a dye, which is 3,4,5 dimethyl thiazole 2-il 2,5 diphenyl tetrazolium bromide, by dehydrogenase to form water soluble formazole. So the MTT, so we have cells with the biomaterials, and after we have left them for interaction for a certain period of time, we then add this MTT dye to it, and if the based on the percentage of active cells, the NADPH dependent mitochondrial dehydrogenase will reduce this MTT into purple formazan. And the formazan are solubilized in solvents like DMSO or isopropanol to determine its concentration using spectrophotometric analysis. So with increasing percentage of metabolically active cells, this uh, color of the purple formazan color will get darker and darker. So, and the color changes from yellow to purple. So based on the color gradient and using spectrophotometric analysis, we can estimate the number of uh, metabolically active cells after interaction with the biomaterial. So the product that is generated during the MTT assay is formazan. Here's the next question. Which of the following biomaterials is not suitable for knee joint replacement? Knee joint replacement implant. Options are scaffolds, polymers, metals, or composites. The correct answer here would be scaffolds. So these scaffolds are basically 3D structures. This scaffolds mainly provide structural support, but then they cannot be used for knee joint replacement implant. Whereas polymers, as shown here, the different classes of biomaterials. Polymers can be used for body parts, joints, and sutures. Then metals, implants, and orthopedics uh, uses metals. So metals can also be used for knee joint replacement. Then we have composites uh, for fracture fixation and joint replacement. So the implants for knee basically should have a smooth surface to prevent friction, and then a strong uh, basic structure for strength and support to the uh, for the leg, or because it is basically it will be a weight bearing structure, so it needs to be able to bear withstand huge amount of strength and prevent any changes in its structure. That's why metal implants are mostly selected. And then these metal implants are also associated with some polymers or composites to get this lubricating surfaces for smooth movements of the joint. But then scaffold itself are structures. They don't have much higher strength to withstand the force of the entire body. That's why they cannot be used for the knee joint replacement. Okay, so that was all the assignment problems for this week, and we'll see more problems, assignment problems in the next day session.